indirect staining and media preparation. So let's start with direct and indirect staining. So what do you know about staining? Staining is done mainly to visualize the bacterial cells, the structure, shape, the morphology and the arrangement of colonies. Before going into staining, we should know that we are employing dyes to stain the cells. And the dyes are usually salts, and they are made up of a positive and negative ion, and one of the ion will be giving you the color. And positive dyes, there are two types of dyes that is called positive dyes and negative dyes. And positive dyes are basic dyes and which is used to stain the cell, bacterial cell. As you all know that the bacterial cells are really negatively charged, even some proteins are also negatively charged. So these positive dyes will combine with the cell wall, negative cell wall, and we could see the color in the microscope, so through the microscope. And the positive dyes are used for direct staining. And some of the examples of direct staining dyes are methylene blue and crystal violet. There are negative dyes, they are called acidic dyes, and as I already said that the cells are negatively charged, so we are adding a negatively charged dye, it will get repulsed. So the background will be colored and you could see the cells colorless. And this type of staining is mainly done to see if a cell has a capsule. So we could see the capsule very much clear than in the direct staining method. And for the example of an acidic dye is like the nitrosin. We will be using nitrosin in this class. And for the direct staining, there is an important step in direct staining method, that is heat fixation. So after pouring the culture onto the slide, we will be heat fixing the slide, because this heat fixing will help the bacterial cells to adhere onto the slide and we could see the cell much more clearer. But we are not using this technique in indirect staining. And the advantage of this direct staining is that we could see the cells very much clearly and easily, but the disadvantage is that we are actually killing the cells, so some kind of distortions can happen to that cells. But in case of indirect staining, the advantage is that there is no heat fixing, and so there is no distortions. And disadvantage is that we should make a smear that is very thin. If the smear is very thick, it may get dried up, you could see cracks in between, so you would not be able to see the cells. So for this class we will be using Micrococcus luteus, and it's not the one that we are using right now. We'll be using E. aerogens. So I'll change that in the slide that you are going to watch in the canvas. So there are two types of cells that we are viewing today. One is the tetrax of cocci, that is round shape, and the single rows, that is rod shaped bacteria. The procedure for direct staining is that we are taking a glass slide and we are with the inoculating loop, we will be adding 2 loopful of the culture onto the slide. We're just making it a thin smear and allowing it to air dry. After air drying, we will be heat fixing the slide, and after heat fixing, we will be adding the dye, that is a methylene blue, onto the top of the smear to cover it. And we are just sitting it like say 30 seconds to like 60 seconds, and then gently rinse the slide with distilled water and allow it to air dry again. But we just want to dry the slides by gently blotting on the sides. You should be careful that you should not wipe the slide. And the procedure for the indirect staining is that in the lab manual and everything, you could see that first we are adding a nitrosin onto the slide, but we recommend to add the culture first and then the nitrosin, because just to avoid the contamination, everything. So first we are taking the glass slide and we are adding 2 to 3 loopful of the culture onto the slide, smear it very well, and then we are going to add the nitrosin. Then just you should be very careful that you should make a thin smear, and then we'll be allowing it to air dry, and then we will be the microscope. We are using the 100x objective, that's an oil immersion objective. I'll be placing a drop of oil onto the slide and we'll be viewing that. Hi guys, now we are going to do the direct staining. For that I'm taking an inoculation tube and I'm gonna sterilize this using this incinerator. I'm keeping it inside, as you guys are familiar with this one. I'm keeping it inside. So now I'm gonna take a glass slide, and gonna add 2 loop full of the culture onto the slide. Just watch out. 1, 2. I'm spreading it as thin as possible, and which will help it to air dry fastly. Okay, and again incinerating my loop. So I'm gonna wait for like 2 to 5 minutes to get it air dried. Then I'm gonna heat fix this. And now you guys could see the slide, it's not still air dried. For easy air drying, you could place the slide on your hand, so the temperature from your hand can make it easily dried up, and after drying up you could add the stain. Now the slide is being air dried. Now I'm gonna heat fix this. So for heat fixing, I'm gluing the slide near the incinerator, keeping like this for some time.
For 30 seconds, I could count the timer.
Yeah, for like 30 seconds. Been heat fixed, and now I'm gonna put the dye. Before that, you need to label. You guys might be confused which side has the actual cells, so you should label it with the wax pencil. So I'm gonna label here like M. I'm gonna stain the slide. So for that, I'm gonna place this slide in the staining rack. See this? And now it's like the time to add the dye, that's a methylene blue. So it should be like 1 to 2 drops is enough. Yep, like this. The slide, make sure you're not forcing too much water on the main part. So after rinsing, I'm just keeping it the slide on a paper towel and gently blotting it dry on the sides, and then gently on the surface. Okay, don't put too much pressure on the center though. And after that, I will be viewing the cells under the microscope. This is a directly stained slide. The cells are colored and the background is colorless. You could see cocoa-shaped cells of M. luteus and rod-shaped cells of Enterobacter aerogenes. And all those purple stuffs are the dyes left after staining. I'll be providing an unlabeled version of this one. This is for you guys to label and submit along with the lab report. Now we're gonna do the indirect staining. For that, the same that we did for direct staining, I'm gonna sterilize the loop and gonna add like 2 to 3 loopful of culture onto the slide. So it's sterilized. So we are using a mixture of the broth that we used in the last class. I think the is cool enough. So now I'm gonna add again like 2 to 3 drops loopful of the culture onto the slide. You could see that I'm just spreading it nicely onto the whole glass slide. Now we're gonna add 1 drop of nigrosin dye onto the slide. Let's see that. Yeah. After adding the dye, I'm gonna spread it very nicely onto the glass slide to make it very thin so that we could see those cells easily and clearly. And this is the slide that we have got, and you could see this part has dried a lot, so I'm gonna place a slide on the microscope and just view what we could find. So uh, first I'll be viewing in 4x, and then I'll be gradually moving on to 10, 40, and only finally I'll be ending up in 100x. So for 100x, as you guys are familiar, we'll be adding a drop of oil and then we'll be visualizing that. This is an indirectly stained slide. Unlike that of direct staining, here the cells are colorless and the background is colored. You could see clusters of cells of M. luteus and single rod shaped cells of Enterobacter aerogenes. That's an unlabeled version for you guys. Just label and submit along with your lab report.